The exosome. When you hear the word exosome, you probably think of the vesicles that are used to transport objects outside of the cell. However, this is not what we're going to be talking about today. Today, we're talking about the RNA exosome, which is used to degrade RNA for processing. So let's begin.
Hi everyone. Today we'll be talking about the exosome, a protein complex that is responsible for degrading the vast majority of RNA within a cell. The exosome is essential for survival, as evidenced by the fact that some variant of it is found in all three domains of life. While eukarya and archaea possess exosomes that are similar in size, structure, and complexity, bacteria also possess a structure called the degradosome that performs the same function. While the exosome does break down nearly all types of RNA, its catalytic activity and its targets do vary slightly depending on its location within the cell. This effect is regulated by a number of cofactors that localize to the cytoplasm, nucleus, or nucleolus. We'll talk about those in depth in just a second, but for now, you can simply think of the exosome as a single entity. Generally speaking, the exosome acts much like the chief operating officer of a company, overseeing the operations that occur on a day-to-day basis and managing any disruptions to workflow. In other words, the exosome kind of calls the shots and decides whether the rest of the machinery in the cell really needs to be there. To better understand why the exosome is so important, let's talk a little more about its substrate, RNA. RNA is a very transient molecule with a typical lifetime ranging from seconds to hours. This process may seem wasteful and inefficient at first glance, but the cell must constantly adapt to changing extracellular and even intracellular conditions. Maintaining this high rate of turnover is critical in adjusting protein expression based on minute environmental differences. This regulation is ultimately what determines a cell's fate. Sensitivity to RNA is also important in detecting foreign particles like viruses, which contain unique identifiers like uncapped 5' phosphate groups and polyuridine motifs. While the viral defense system is most responsive or has the highest affinity to these elements, having excessive amounts of endogenous RNA can also overwhelm its ability to distinguish between good and bad RNA, potentially leading to an autoimmune response. Lastly, having too much free-floating RNA may also pose long-term consequences such as irreversible DNA damage. As you can see in this figure, DNA strands that base pair with spliced RNA rather than their anti-parallel counterpart can actually form structures called R-loops. These R-loops can undergo double-strand breaks, and the unpaired regions are simply discarded at the next round of replication. Obviously, the loss of entire chunks of DNA is disastrous to the cell.
Now, because the exosomes are heavily involved in the maintenance of RNA structures, a quick rundown of transcription is in order before we proceed. RNA arises from DNA with the use of RNA polymerase during transcription. Specifically, RNA polymerase reads DNA from the 3' end to the 5' end to create RNA transcripts that run from 5' to 3'. Different RNA polymerases create different RNA structures. RNA polymerase 1 synthesizes ribosomal RNA, or rRNA. RNA polymerase 2 synthesizes heterogeneous nuclear RNA, or hnRNA, as well as small nuclear RNA, or snRNA. And RNA polymerase 3 synthesizes transfer RNA, or tRNA. After transcription, some RNA can undergo post-transcriptional modifications before becoming fully usable. This is where the exosome comes in handy. The RNA exosome aids in these modifications by degrading mRNAs lacking stop codons, mRNAs containing premature stop codons, and also helps in the degradation of mutant, aberrant, or viral RNA. Take, for instance, how hnRNA undergoes many modifications before it becomes usable mRNA. These modifications include alternative splicing and the removal of non-coding sequences or introns, the addition of a 5' cap and 3' poly A tail to prevent rapid degradation, and, in some cases such as for the mRNA, methylation that regulates localization, degradation, and gene silencing. With all this stated, overall the exosomes perform cellular quality control, post-transcriptional processing, and assists in RNA turnover and maintenance. It does so by acting as a 3' to 5' exonuclease and endonuclease, meaning that it can operate with RNA sequences both deep within the polynucleotide chain and at either end of the RNA strand. I should stress one final time that this is the RNA exosome, not to be confused with the extracellular transport vesicle system of the same name. Just as a quick overview of what's to come, the exosome is composed of 10 essential subunits. A useful mental image to keep in mind is a trash can or shredder that degrades problematic RNA that gets filed into it. Mutations in this machine lead to longevity-related disorders such as melanoma and premature aging syndrome.
The human exosome is made up of 9 catalytically inactive subunits and 1 active subunit. 6 of these catalytically inactive subunits form a ring. In addition, 3 other subunits sit on top of the ring and form what we call a cap. Although it is called a cap, it does not necessarily act like one, because all 9 subunits form a tunnel where RNA is funneled through to eventually be degraded by the active ribonuclease DIS3 or DIS3L. The difference between these two active ribonucleases depends on whether the exosome is located in the nucleus or the cytoplasm. The nucleus exosome has also been shown to have one extra subunit called ExoC11, which attaches to the cap of the complex. Lastly, and very importantly, the exosome complex has been shown to have several cofactors associated with it. As we mentioned earlier, the RNA exosome works a little bit like a trash can shredder. Just as a trash can shredder shreds paper, the RNA exosome is able to shred RNA. But in order to do this, it needs specific RNA cofactors, such as adapters, which aid in the binding and targeting of RNA to the exosome. Other cofactors, such as helicases, disrupt RNA structures and prepare it for processing and degradation. Once this is done, the single-stranded RNA can be threaded through the central channel of the exosome we talked about earlier. Once it reaches the catalytically active subunit, DIS3, it can be degraded in the 3' to 5' direction. One important thing to note is that different cofactors are needed depending on where the exosome is located and whether processing or degradation is being performed. But that is all a part of the regulation of the RNA exosome.
The regulation of the exosome is all about the cofactors. The cofactors associate with the 3 subunit cap of the exosome and can have different effects, such as the ones discussed in the previous section. The necessary cofactor is dependent on the location of the exosome and the desired type of activity. One example of this is aberrant tRNA degradation in the nucleus. For this situation, the TRAMP complex associates with the exosome. This complex contains a specific RNA adapter, helicase, and poly-A polymerase. Another example is mRNA quality control, or ribosome stress response in the cytoplasm. The SCI complex associates with the exosome and contains a helicase, a scaffolding protein, RNA adapters, and exosome adapters. The exosome requires many cofactors to function properly and does not work without them.
From a medical standpoint, there have been 7 mutations in the exosome that have been found to cause 4 distinct disease phenotypes. These 4 diseases are both incredibly uncommon and very serious, more often than not proving to be fatal. Trichohepatoenteric syndrome is a disease which affects the hair, liver, and intestines. This disease, which affects 1 in 1 million people in the U.S., is characterized by a woolly hair phenotype and also tends to present in infants as syndromic diarrhea. As a result, total parenteral nutrition is required, which means food must be delivered intravenously to bypass the faulty GI system. Liver failure is another common symptom. Just over 50% of patients with trichohepatoenteric syndrome have intellectual disability associated with the disease. The disease is caused by mutations in either the SCI-VL2 or TTC37 genes, but the molecular pathology is not yet understood. No proper antibody response has been observed to both infections and vaccines, which makes trichohepatoenteric syndrome almost always fatal in the first few weeks of life. However, some do survive into adulthood. Premature aging syndrome is a disease more recently associated with mutations in the exosome. Individuals with this disease appear phenotypically normal at birth, but begin to age rapidly throughout the first few years of life. First characterized by early hearing and vision loss, premature aging syndrome arises from a mutation in the EXO-SC2 gene. Individuals with premature aging syndrome are typically short in stature and often have mild intellectual disability. While other premature aging syndromes, such as progeria, exist, this specific condition associated with the exosome was discovered in 2016, so not much is known about it. Yet all the patients involved in the research at the time of the study are still alive today, the oldest being in their mid-40s. Pontocerebellar hypoplasia is a neurodegenerative disorder characterized by muscle weakness and developmental delay. This condition arises from mutations in either the EXO-SC3, 8, or 9 genes. This disease causes different parts of the brain, namely the cerebellum and pons, as the name suggests, to form much smaller than normal. The condition becomes noticeable as the brain growth continues to be slow throughout infancy and early childhood. Individuals with pontocerebellar hypoplasia typically do not survive past the first 2 decades of life, with many passing away in infancy. Currently, only symptomatic treatments exist. Multiple myeloma, a plasma cancer, arises from a mutation in the DIS3 gene. Multiple myeloma results in bone weakness, as it increases generation of osteoclasts, which break old bone down, without generating osteoblasts, which form new bone. Additionally, as multiple myeloma is a plasma cancer, compromised plasma cells lead to reduced antibody production, leaving patients more susceptible to infections. Some patients have experienced kidney damage and even kidney failure due to a harmful antibody produced by the myeloma cells. Treatments include chemotherapy, radiation, and stem cell transplants. As with most cancers, if caught early, patients may have a good chance of survival. However, on average, the 5-year survival rate is 50.7%.
The consequences of mutated exosomes are dire. We have established that as much. The different exosome changes impact different tissues altogether. It was once assumed that every type of exosome mutation has similar effects, but a targeted impact on one part of the body is now evident with a multitude of diseases popping up over decades of clinical studies. Specific residue mutations can allow different degrees of organism vitality overall, but with low function. The 2 most common mutations are missense, resulting in a change in residue, and nonsense, causing a full stop in translation. The latter deletes important information and instead is the reason for loss of function in the exosome.
The RNA exosome is a complicated complex with as many as 11 different subunits and a plethora of cofactors necessary for full functionality. This complex is vital to the functioning of our cells. Mutations in this complex and its associated pathways are often fatal. That said, the complex is also responsible for some of the many things that allow us to be living, complicated beings, such as cell differentiation, removal of viral RNA, and protection from DNA mutation.